All right. Hi, everyone. My name's Matthew. I'm a postdoc at the University of Michigan, and I'm really excited to have the opportunity to talk to you about some recent work looking at how we can extend our ability to understand the functional structure of evolved genotypes. How and why complexity evolves is a key open question in evolution. However, beyond that, it's also an open question how we should define and conceptualize complexity. There's a lot of different complementary approaches that have been used to try to understand complexity, including phenotype-based approaches where you remove phenotypic traits and see what's adaptive, looking at particular biological processes like development and ecology, as well as looking at phenomena outside of evolved genotypes like cultural and social transmission. In this particular talk, we'll look at one specific complexity measure, sequence complexity, which tries to quantify the number of sites in a genome that are adaptive and contribute to fitness in an evolved organism. A typical approach to measuring sequence complexity uses site-by-site knockouts to count the number of sites that contribute. So to get a sense for what this means, we can analogize a genome we're interested in analyzing to a Jenga tower, and then our site-by-site knockouts are removing one block at a time and trying all the different blocks in the tower and seeing how many blocks cause a detectable fitness outcome in this analogy, the Jenga tower collapsing. So a sequence complexity measure can be very informative, especially if we take it over time during an evolutionary simulation. I have some example data here from a project I was involved in a few years ago looking at evolution of phenotypic traits in simulated multicells. Here I have evolutionary time on the x-axis and a sequence complexity measure on the y-axis. It was generated using site-by-site knockouts, and different phenotypic traits are color-coded on this diagram. And so by analyzing sequence complexity, we can get a sense for the trajectory of evolution in this particular simulation, where we see sequence complexity increasing over maybe the first half of this simulation with some interesting discontinuities, and then decreasing slightly over the second half as perhaps there's some kind of selection for parsimony or mutational robustness. However, when we were working with this system, we started to wonder the extent to which this site-by-site knockout process was giving us a complete picture about the functional structure of our evolved genotypes. And so we did some additional experiments where we returned to the sites that had been detected as neutral in our original site-by-site knockouts and started sampling groups of these sites and knocking them out all together. And so on the x-axis of this diagram, I've got the number of sites that get grouped together and knocked out, and then on the y-axis, the fitness outcome. And you can see the significant fitness outcomes are color-coded orange. And when we start sampling even a modest number of these ostensibly neutral sites and taking them out together, we start to see a fitness effect. So it appears that these have some kind of contribution to the functionality of the genotype, but we're not able to detect them through site-by-site knockouts. And so we term these sites that aren't detectable through site-by-site knockouts as cryptic sequence complexity. And the goal of this current work is trying to develop methods to be able to systematically quantify and characterize these particular this aspect of complexity. In this presentation, we'll talk about two different reasons why sites might manifest as cryptic due to redundancy effects and additive small effects. We'll describe both of those kind of in more detail shortly, and then we'll overview three different proposed approaches to try to quantify different aspects of this cryptic complexity. So To talk about kind of the first mechanism through which sites might manifest as cryptic, we'll return to our Jenga metaphor and talk about redundancy. I think most people who've played a game of Jenga are kind of familiar with the concept of the blocks within a single layer of the tower being functionally redundant, where you can take out any one block and not have a detectable fitness effect. But these blocks nonetheless are contributing to the structure of the tower, because if you take out all three, you see a catastrophic effect. So that's kind of the first mechanism through which sites might manifest as cryptic. The second mechanism is additive small effects. And to discuss this, we'll extend our metaphor a little further, perhaps maybe a little further than we should. And we'll suppose that our Jenga tower is additionally adorned with glittery sequins. And perhaps in this analogy, these sequins have some kind of protective effect against predation on the Jenga tower. But in this, just to be clear, kind of this extended analogy, we're considering each of these little sequins as a genome site, and we're considering the possibility that they might have a small additive effect in protecting against predation, or they might be entirely neutral. And so because the contribution of any one sequin on our tower here is small with respect to fitness, right? If we just take one sequin off, we might not be able to detect a clear change in the fitness of the tower. But if we take lots of sequins off, we might lose enough of that protective effect to start to be able to detect significant fitness changes from this genome. So now that we've defined these two mechanisms through which cryptic sequence complexity can manifest, we'll now move to talking about kind of some of the proposed approaches in this extended abstract to going after quantifying and characterizing this type of structure. So the first approach we'll talk about is dose knockouts. This kind of extends a little bit of what we were talking about earlier, building on the fact that kind of the intuition that if we have additive small effects and we take off one side at a time, the majority of, we're very likely to not see a detectable fitness effect. But if we sample large numbers of sites and take them all out, we're likely to sample enough sites that are having some small effect that we'll see a detectable fitness effect. And so what we can do is try to interpolate between these two extremes with taking out different numbers of sites at a time, and use that process to try to characterize the functionality of sites. So specifically, what we'll expect to see when we increase the number of sites that we're sampling is a dose response curve, where along the y-axis, we have increasing probability of detecting a significant fitness outcome. And we can model each individual knockout as a coin flip event, where there's a probability that we might see a small additive effect, or that we might see no effect at all if we happen to sample a completely neutral site. And we can consider kind of our dose knockouts as a sequence of coin flips, where in order to see a phenotypic effect, in order to see a fitness effect from this dose knockout, we need to get enough successes in our coin flips to build up enough small effect sites to reach the threshold of detectability. And it turns out that this kind of, if we pose a scenario this way, we can draw a relationship between the sequence of coin flips and the negative binomial distribution, which gives you the waiting time until you have a certain number of successes in a sequence of coin flips. And this dose response curve happens to be congruent to the cumulative distribution function of negative binomial distribution in this scenario. And so what we can do is use experiments to generate this dose response curve and then fit a negative binomial distribution to it and estimate the two parameters of the negative binomial distribution, P, the probability of any individual coin flip being a success, and N, the number of successes required in a coin flip sequence. And estimating P will tell us about the fraction of sites in our genome that are contributing small additive effects. And estimating N will tell us about how many of those effects we need to stack up in order to reach the threshold of detectability. So that will tell us about the effect size of individual sites. The next approach that we're proposing is trying to go beyond just looking at these additive small effect sites and more holistically detect both additive sites and sites that might be contributing to cryptic sequence complexity because of redundancy. What we propose to do here is a sequential knockout process where we start with the wild type genome and we start trying knockouts on it. And once we detect a site that we can remove from the wild type genome without causing a detectable fitness effect, then we continue doing knockouts on that genome. And basically, just like playing a game of Jenga, we pull blocks out of the tower until we reach a minimal configuration where there's no blocks left that we can pull out without having a detectable fitness effect. And in the presence of cryptic sequence complexity, it turns out that there are actually usually many different possible configurations of kind of a minimal structure. Just like when you play a game of Jenga, you can end up with all kinds of different end state tower configurations. So we can think about sampling over this distribution of possible skeletonized genome structures and then analyzing the structure of those genomes that we've sampled through knockouts to learn more about the cryptic structure. So what I have plotted here is different sampled skeletons on the x-axis and different sites within the genome on the y-axis. And there's if there's a site that's truly neutral in our genome, we expect it to be removed from all the skeletons. So here removal is indicated by a red X. And so the site in the first row is neutral and we've removed it from all of our skeletons. Sites that are critical to fitness should show up in all of our skeletons because they, by definition, can't be removed without seeing a deleterious fitness effect. And then our cryptic sites, whether additive small effects or redundant, will show up as being preserved in some skeletons, but removed in other skeletons. And so by sampling multiple skeletons, we can start to kind of identify these cryptic sites and kind of start keeping account of the number of distinct sites that we're seeing across all of our sampled genomes. And it turns out with a little bit more statistics, we can actually get a little bit more information out of this process, where if we extend our analogy to kind of think of each of these individual sampled skeletons as a sampling event in like an ecological mark recapture experiment, we can, just like ecologists use mark recapture experiments where they sample, they go to the field and they sample critters, and by seeing the frequency at which they recapture critters across different samples, they're able to estimate the overall size of the population of critters. We can think of sampling cryptic sites and the frequency at which these cryptic sites recur across our skeletonized genomes. We can use that to estimate the overall size of the population of cryptic sites, and even kind of estimate the presence of sites that we didn't detect directly within our sampled genomes. And there's some statistics that's necessary to take into account here. One really important factor being the fact that sites very well may have different probabilities of ending up in skeletons. But it turns out that ecologists have really, really well-developed statistics for this type of mark recapture sampling, including taking into account the fact that some critters are more trap shy, they call it, are more difficult to capture. And so we can apply some of those extended mark recapture statistics to get hopefully an accurate estimation of the overall quantity of cryptic sites that occur. So our last technique that we'll overview very briefly here is just a little extension on the skeletonization process to get a little bit more information out, where when we skeletonize a genome, we reach a minimal configuration where we expect to see a detectable fitness effect from further knockouts. So we can go ahead and do those site-by-site knockouts on our skeleton, and we'll expect to see a fitness effect every time. And the magnitude of those fitness effects can tell us about the type of cryptic sequence complexity that we're capturing within these skeletons, where if we see mild effects, we can deduce that those sites were contributing in a small additive way. And if we see large critical fitness outcomes, we can conclude that those sites were probably involved in some kind of redundancy. And when we cross the redundancy threshold, we see a big effect. So to wrap up here, we covered a lot, so I'll just briefly recap it. We defined cryptic sequence complexity as sites that are contributing to the functionality of a genome but aren't detectable through individual knockouts. We talked about 2 reasons why sites might be cryptic: because of additive small effects and because of pleiotropic redundancy. And we used a Jenga metaphor to get some intuition for those 2 possibilities. And then we introduced 3 different techniques to try to characterize and quantify this type of cryptic structure within a genome. This is really preliminary work, kind of proposing these methods, and so there's a lot more left to do to really robustly test them on genomes of different structures where we know the ground truth and see their capability. See the capability of these methods to hopefully recover accurate information about the ground truth. And it'll be really important also to compare these types of approaches to simpler approaches like just doing 2-site knockouts, and see how much of cryptic complexity is just captured by doing those simpler approaches. There's some open questions left in figuring out the optimal way to run some of these experiments, in particular figuring out how to pick the dosages for our knockouts to get the maximum information for each knockout trial that we do. And I think it's also an interesting problem trying to figure out how and if we can parallelize the skeleton generation process, because that's potentially a lengthy multi-step process. Another thing that we'll have to figure out how to deal with is the fact that in many systems, we have to test fitness by doing competition experiments, which can be stochastic. And so dealing with the fact that we might have stochastic false positives and false negatives, and if we do the same knockout trial twice, we might get 2 different results. That will add some further complications to these proposed methods. The last complication and piece of future work that I'll highlight is the complexity of and kind of the difficulty to successfully orchestrate of this type of workflow, where we're using the results of previous knockout experiments to determine what knockout experiments we should do next. And so you can't just specify all of the trials that you need to perform ahead of time and submit all of those jobs to do those experiments all at once. You have to have kind of this recursive workflow where you get information from the knockout trials and incorporate it into your orchestration system, and then use that to do more knockout trials. And figuring out a reasonable way to do this that generalizes across A-life and digital evolution systems will be really important. With that, I will acknowledge my contributors, including Gertrude the cat, who was featured on the slides, and I'll take questions. Thank you.